Hi, welcome to part 2 of our nutrition and metabolism lectures at Wayne County Community College. We're going to review our beginning of glycolysis and then move on into the tricarboxylic acid cycle and then on to the electron transport chain in this series of 30 slides. We'll also do some practice questions at the end. Okay, so we started out with glucose transport into the cell. As you'll recall, glucose was phosphorylated using ATP by the enzyme, either hexokinase or glucokinase, depending on the cell. And it was phosphorylated to G6P, which trapped it in the cell by giving it a negative charge. The phosphorylation of glucose prepares it for further metabolic reactions in the cell. And this is irreversible in almost all the cells except for those of the intestinal mucosa, importantly in the liver and in the renal cortex. So now that glucose has been phosphorylated, it is ready to enter glycolysis. And glycolysis is a series of reactions that will extract the energy from glucose by splitting it into two 3-carbon molecules called pyruvate. Glycolysis is an ancient metabolic pathway which evolved a long time ago, so therefore it is found in a great majority of organisms that are alive today. In organisms that perform cellular respiration, such as we humans, glycolysis is the first stage of this process. However, glycolysis itself does not require oxidation, and so it will be used in both many anaerobic organisms, those that don't use oxygen, as well as in organisms that do use oxygen. It just can occur under anaerobic conditions. It also occurs in the cytoplasm of human cells. Okay, so let's look at glycolysis. So this course in anatomy and physiology is more of a practical course for those of you going on into clinical aspects of medicine. But if you were really interested in getting into the weeds of the biochemistry, glycolysis has 10 major steps, and you can trace them through to your delight if you'd like. But we're going to look more at the greatest hits and the overall results of glycolysis. So glycolysis has two basic main phases, an energy requiring phase and an energy releasing phase. During the energy requiring phases, our carbon skeleton is going to get split into 3 carbon units, and it will get phosphorylated, using up some ATP. So this is where we have an initial investment of 2 ATP to phosphorylate our 3 carbon sugars. And then we will enter our energy releasing phase, in which each 3 carbon sugar will be converted into another 3 carbon molecule called pyruvate through a series of reactions. In these reactions, 2 ATP molecules and 1 NADH, the reduced form of a coenzyme, are made. Because this will take place twice, one for each of the 2 3-carbon sugars, it will make overall 4 ATP and 2 NADH. Since we had our initial investment in the energy requiring phase of 2 ATP, we come out of glycolysis with forming an overall net 2 ATP. And we will form 2 reduced coenzymes, NADH. And we will have 2 3-carbon pyruvates. So let's look at our overall slide for a second. Okay, so overall, glycolysis converted our 1 6-carbon molecule of glucose into 2 3-carbon molecules of pyruvate. The net products of this process are the 2 molecules of ATP that are produced and 2 molecules of NADH. This occurred in the cytoplasm of the cell. We're now going to be ready to enter into the Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle. So we will move our products into the mitochondrion, And there we'll go through the citric acid cycle, where we'll want to pay attention to the formation of coenzymes, reduced coenzymes, NADH and FADH2, which will eventually enter the electron transfer phosphorylation. And that's where all the ATP will be made. We'll only make 2 more ATP in the citric acid cycle. Okay, so let's start looking at our citric acid cycle. At the end of glycolysis, our glucose, which was a 6-carbon sugar, was broken down into 2 3-carbon molecules of pyruvate. And that pyruvate still contains a lot of extractable energy. So pyruvate oxidation is the next step in capturing the remaining energy in the form of ATP, although no ATP is directly made during the pyruvate oxidation stage. In eukaryotes, this step will take place in the mitochondrial matrix, the innermost compartment of the mitochondria. In prokaryotes, like most of our bacteria, it will happen in the cytoplasm. Overall, pyruvate oxidation is going to convert that 3-carbon molecule of pyruvate into acetyl-CoA, which is a 2-carbon molecule attached to the coenzyme A. When we form this new 2-carbon acetyl-CoA molecule, we will release 1 molecule of carbon dioxide for each of the pyruvates that entered into this. And we will produce a reduced coenzyme of NADH. The acetyl-CoA will end up acting as the fuel for the citric acid cycle in the next stage of cellular respiration. Okay, so let's look at what happens at the end of the tricarboxylic acid cycle. Ultimately, from our 2-pyruvate, we will release 6 carbon dioxide molecules and 6 molecules of water. Our pyruvate, since we've had 2 of them, will undergo 2 rounds of the citric acid cycle, and we will generate a bunch of reduced coenzymes. 6 NADH, it was the NAD plus the oxidized form, that will be reduced and contain all that extra energy as NADH. And FAD will be reduced to 2 FADH2. We will also, through the turn of the citric acid cycle, produce 2 ATP. So let's look at a little bit of these stages more closely. Okay, so here's a scary cartoon. We already went through glycolysis and ended up with our pyruvate. And now we're going to enter the second stage of aerobic respiration, which is known as the tricarboxylic acid cycle, or the citric acid cycle, or the Krebs cycle. All those terms can be used interchangeably. Okay, so we're going to start out with our pyruvate at the top of this cartoon. The thing I want you to pay attention to throughout this turning through the citric acid cycle is that just before we enter the citric acid cycle, you'll see that we make some NADH, our reduced coenzyme. That's a high energy coenzyme. Okay, we'll also make more NADH throughout the turns of the citric acid cycle and FADH2. You'll see those in those yellow colored boxes throughout this cartoon. Also at the bottom of this cartoon, we'll make some either ATP or GTP. Okay, we make a minimal amount of ATP or GTP through the turn of the citric acid cycle. Our money is going to be from making all those reduced coenzymes that will later enter oxidative phosphorylation or the electron transport chain. Okay, so let's get to it. Don't obsess about the carbon skeletons and the changes from oxaloacetate to succinyl-CoA to back to citrate, okay? Focus on the reduced coenzymes in the formation of ATP. All right, so let's go through this scary slide. All right, so we're starting out with our pyruvate. Okay, and our pyruvate was made out in the cytosol. Our 6-carbon glucose was split into 2 3-carbon pyruvates out in the cytosol. Our pyruvate molecule is then going to be converted into carbon dioxide plus a 2-carbon acetyl group, which will become attached to coenzyme A, forming acetyl-CoA, which is another activated carrier molecule that will enter the citric acid cycle. The oxidative breakdown of our food molecules will take place entirely in the mitochondria. Here, the acetyl group in acetyl-CoA, which is linked to that coenzyme A through a high energy linkage, will be easily transferable to other molecules. So that's the changes in the carbon skeleton. It'll start out with being transferred to the 4-carbon molecule oxaloacetate, and the acetyl group enters a series of reactions called the citric acid cycle, or the Krebs cycle, or the tricarboxylic acid cycle. The acetyl group is oxidized to carbon dioxide in these reactions, and large amounts of the electron carrier NADH will be generated. The high energy electrons from NADH will eventually be passed along the electron transport chain within the mitochondrial inner membrane, and we'll get to that later. That's where most of the ATP will be formed. You see at the bottom, we also form 1 ATP here. The energy to drive ATP synthesis in the mitochondria ultimately derives from this oxidative breakdown of the food molecules. The phosphorylation of ADP to form ATP is driven by the electron transport in the mitochondria known as oxidative phosphorylation. And we'll get to that later, all the events that occur within the mitochondrial inner membrane. The citric acid cycle accounts for about 2/3 of the total oxidation of carbon compounds in most cells, and its major end products are carbon dioxide and the high energy electrons in the form of NADH or FADH2. Okay, so let's summarize our tricarboxylic acid cycle. The citric acid cycle, which takes place inside the mitochondrial matrix in eukaryotic cells, results in the complete oxidation of the carbon atoms of the acetyl groups in acetyl-CoA, converting them to carbon dioxide. The acetyl group is not oxidized directly. It instead transfers the acetyl from the acetyl-CoA to larger molecules. First, the 4-carbon molecule oxaloacetate to form the 6-carbon tricarboxylic acid, citric acid, for which the cycle is named. The citric acid molecule is then gradually oxidized, allowing the energy of this oxidation to be harnessed to produce energy-rich activated carrier molecules. This chain of reactions forms a cycle because at the end, the oxaloacetate is regenerated and enters a new turn of the cycle. So in summary, each turn of the cycle produces 2 molecules of carbon dioxide, which are waste products, plus 3 molecules of the reduced coenzyme NADH, and 1 molecule of either GTP or ATP, and 1 molecule of the other reduced coenzyme, FADH2. Like NADH, FADH2 is a carrier of high energy electrons and hydrogen. As we'll get into pretty shortly, the energy that's stored in the readily transferred high energy electrons of NADH and FADH2 will be utilized subsequently for ATP production through the process of oxidative phosphorylation, the only step in the oxidative catabolism of foodstuffs that directly requires gaseous oxygen from the atmosphere. So let's now move in to the electron transport system. It is in this last step of the degradation of the glucose molecule that the major portion of its chemical energy is released. In this final process, the electron carriers NADH and FADH2 transfer the electrons that they have gained when oxidizing other molecules to the electron transport chain. The electron transport chain is embedded in the inner membrane of the mitochondria. As the electrons pass along this chain of specialized electron acceptor and donor molecules, they fall to successively lower energy states. The energy that the electrons release in this process will be used to pump H+ ions, or protons, across the membrane from the inner mitochondrial compartment to the outside. A gradient of hydrogen ions is thereby generated. The gradient will serve as a source of energy, being trapped like a battery to drive a variety of energy requiring reactions. The most prominent reaction, the one that we care about, is the generation of ATP by the phosphorylation of ADP. Okay? So remember that in the electron transport system, these high energy electrons, along with their protons that were removed during the tricarboxylic acid cycle, enter this chain of molecules that are embedded in the inner mitochondrial membrane. And as those electrons will move down the chain, they release small bursts of energy to pump protons between the inner and outer membranes of the mitochondria. So let's look at a cartoon that talks about the generation of ATP by ATP synthase at the inner mitochondrial membrane. Here, the generation of an proton gradient across the membrane by the electron transport reactions will take place. A high energy electron from the oxidation of a metabolite will be passed along sequential carriers to lower energy states. At the end of this series of electron transfers, the electrons are passed to molecules of oxygen. So oxygen is the final electron acceptor. That's very frequently a test question. Electron is the final electron acceptor in the electron transport chain. Okay, so as I was saying, the electrons are passed to molecules of oxygen gas that have diffused into the mitochondrion, which simultaneously combine with protons from the surrounding solution to produce molecules of water. The electrons have now reached their lowest energy level, and therefore all the available energy has been extracted from the food molecule that was oxidized, from the glucose that we started out with. This process is termed oxidative phosphorylation, and it occurs in the plasma membrane of bacteria as well as the inner mitochondrial membrane of eukaryotes. So let's now summarize what happened throughout the whole sum total of aerobic respiration. Okay? In total, the complete oxidation of one molecule of glucose to water and carbon dioxide is used to produce 2 ATP in glycolysis and 2 NADH in glycolysis. In the tricarboxylic acid cycle, we formed acetyl-CoA, and ultimately, from that, we garnered 6 NADH and 2 FADH2 and 2 ATP. And then, throughout the electron transfer phosphorylation, we formed 32 ATP. Now, just to get a couple of terms straight that you might hear, remember that the electron transport chain is a series of proteins and organic molecules that are found in the inner membrane of the mitochondria. The electrons are passed from one member of the transport chain, and we're not going into the in-depth of all those members, into another series of oxidation reduction reactions, redox reactions. The energy released in these reactions will be captured as a proton gradient, which is then used to make ATP in a process called chemiosmosis. So when you see the word chemiosmosis, that's sort of that final stage where you're actually generating the ATP. So together, the electron transport chain and chemiosmosis make up oxidative phosphorylation. And that's how to keep those three terms of chemiosmosis, electron transport chain, and oxidative phosphorylation straight. Okay, so let's summarize again some of our carbohydrate metabolism. Oxidative phosphorylation is the joining of a phosphate group to ADP to form ATP by the action of the enzyme ATP synthase. And as I just said, oxidative phosphorylation is made up by together the electron transport chain and chemiosmosis. Recall that we started all of this with glycolysis, which was an anaerobic pathway. In glycolysis, we broke down sugar, a 6-carbon sugar of glucose, into the 3-carbon molecule of pyruvic acid or pyruvate. And a series of reactions associated with glycolysis are necessary for both anaerobic and aerobic pathways to work, because ultimately that end product of the anaerobic glycolysis will end up going through our tricarboxylic acid cycle and oxidative phosphorylation to efficiently produce ATP for our cellular metabolism. Soon we will also look at what continues to happen under anaerobic conditions with the pyruvate being reduced to lactic acid through a process known as fermentation, which is that little side arrow on the slide with all of the coins or the currency of ATP building up. We'll get to that in a little bit. Okay, so now let's just sort of summarize our cell machinery that we've learned so far for the glucose catabolism. Let's look at this next slide that has a nice cartoon summarizing everything. So we started out with glucose that came from our food through that alimentary canal, that tube inside of us, so it's the outside world, was part of digestion that we took up through our cells at the brush border. Glucose enters the cell through a glucose transporter, and it's immediately phosphorylated to G6P by either hexokinase or glucokinase utilizing ATP. So that's our investment stage of ATP. The glucose, which is a 6-carbon sugar, is eventually broken down into pyruvate. Okay, so at the end of glycolysis, we have 2 pyruvate molecules that still contain a lot of extractable energy, and they're still out in the cytosol where the glucose entered through that glucose transporter. So the oxidation of pyruvate is the next step in capturing the remaining energy in the form of ATP, although no ATP is directly made during pyruvate oxidation. Okay, so now this step takes place in the matrix of the mitochondria, the innermost compartment of the mitochondria. Unless, of course, it's in prokaryotes where it continues to happen in the cytoplasm. Pyruvate oxidation will convert that pyruvate, which is a 3-carbon molecule, into acetyl-CoA, a 2-carbon molecule that's attached to coenzyme A, producing the reduced coenzyme NADH and releasing 1 carbon dioxide molecule in the process. The steps of pyruvate oxidation are carried out by a large enzyme complex called the pyruvate dehydrogenase complex, which consists of 3 interconnected enzymes and includes 60 subunits. So just remember pyruvate dehydrogenase complex. The acetyl-CoA then enters the citric acid cycle which is the central driver of cellular respiration. So the acetyl-CoA produced by the oxidation of pyruvate, originally derived from the glucose, will go through the citric acid cycle, which is a series of oxidation reduction reactions, and we will harvest much of the bond energy in the form of the reduced coenzymes NADH and FADH2 and ATP or GTP molecules. The reduced electron carriers NADH and FADH2 that are generated in the carboxylic acid cycle will pass their electrons into the electron transport chain, and through oxidative phosphorylation, they will generate most of the ATP that is produced in cellular respiration. Okay, so let's now look at the energy that was extracted from our single glucose molecule. Our 1,6-carbon glucose enters the cell and undergoes glycolysis, at which time we net 2 ATP, but we also produce 2 NADH. Those 2 NADH will go on through the tricarboxylic acid cycle and oxidative phosphorylation to produce 4 more ATP. Then, in oxidizing our pyruvate to acetyl-CoA, we manufacture 2 more NADH, and that will go on to form 6 more ATP. We then, through the citric acid cycle, form 2 either ATP or GTP, and will form 6 more NADH, which will become 18 ATP in oxidative phosphorylation. Our 2 FADH2 will then go on to form 4 ATP. So from that single molecule of glucose, we generate 36 total ATP. Now, most current sources actually estimate that the maximum ATP yield for a molecule of glucose is really around 30 to 32 ATP. The range is lower than previous estimates or the theoretical amount because it accounts for the necessary transport of ADP into and ATP out of the mitochondrion. Okay, so let's move on to some practice questions. Maybe you want to take a little break first, and then we'll see what it is we've actually learned. Okay, so now for some practice questions. Let's go to the first one. In which of the following cells is glucose phosphorylation reversible? So think about this, maybe hit pause as we go through the questions, and then we'll move right on to the answers. Okay, so the heart, the pancreas, the liver, the muscle, or the adipocytes. So recall, in the liver is where we have the phosphorylase, and it can actually reverse that reaction. In most cells, that doesn't happen. So the liver, the kidney cortex, and in some of the intestinal cells is where you'll see this happening. Okay, so which of the following statements is not true? I'll give you a chance to read through all these and let you decide. Maybe hit pause, and we'll go on through this. Okay, so the answer is D. Glycolysis requires the use of oxygen. Remember, glycolysis is an anaerobic process. Although it is an essential process in preparing glucose for the aerobic process, later on the citric acid cycle and the electron transport chain / oxidative phosphorylation. Okay, let's go on to our next slide after this. Okay, where does the citric acid cycle take place? Is it in the ribosome, the cytoplasm, the mitochondrial matrix, the inner mitochondrial membrane, or the thylakoid membrane of the chloroplast? Okay, so the citric acid cycle takes place in the mitochondrial matrix. Remember, protein synthesis is what occurs on the ribosome. Glycolysis occurs in the cytoplasm. And the inner mitochondrial membrane is the area for the electron transport chain, as is the thylakoid membrane of the chloroplast in some cells, some eukaryotes. Okay, so our answer is the mitochondrial matrix. Just remember the site of all these. It's very highly tested. All right, next question. Which product of pyruvate oxidation enters the citric acid cycle? Okay, do you remember what happens? Okay, so pyruvate is our 3-carbon fragment, and it has carbon dioxide split off, so that is not what enters the citric acid cycle. That's a waste product. And our 2-carbon fragment, or acetyl group, joins with CoA and forms acetyl-CoA. So that is the answer. Remember, you want to remember why the right answers are correct, but also know why the wrong answers are incorrect, and you'll do a lot better on your exams that way. Okay, next question. The reduced coenzymes NADH and FADH2, along with ATP, are important products of the tricarboxylic acid cycle, or the Krebs cycle, or the citric acid cycle. Okay, so you should all know the answer to this question. This is an obvious, true answer. Okay, those are those important products. Okay, let's look at what the end of glycolysis result is on the next slide. So what is the end result of glycolysis? Well, remember, we made pyruvate, we made 2 ATP, and we also reduced some NAD plus to NADH. So our answer is C. The answer is C. Glycolysis produces ATP, pyruvate, and the reduced coenzyme NADH when it oxidizes glucose. Okay, let's see if we know anything about the electron transport chain. So which of the following is true about the electron transport chain? Okay, think about it a moment. Pause the video for a second. And the answer is that the movement of the electrons down the electron transport chain drives the formation of a proton gradient. And we just went over that at the end of our slides, so I'm pretty sure you got that one correct. Okay, good luck, and I'll see you for the remainder of this chapter in the next set of videos.